So let's start. Now I am showing two of the jail picture. This jail picture actually I have downloaded from the internet, and this jail picture for SSR marker. Here we can see there are two types of band here. Okay, this one is one type band, another one is the below. Maybe it's a 150 base pair, and this is 120 base pair. Okay, maybe. So this type of band we have all over seen in the papers, or when we are doing in jail, then we can found jail electrophoresis, then we can found this type of picture. Now how can convert it this type of picture in binary matrix? What are the importance of the SSR marker overall the dominant marker like RAPD? So this is the SSR marker, and this is the picture of RAPD marker. Here you can see multiple number of bands. So in SSR there is band range is low, and you can found some specific bands. But in RAPD you can found number of bands. Okay. So from that, how can we score the actually in binary format? For the scoring, we need to identify. Let's start from the SSR marker. So here you can see this is the band, maybe the 200 between the 200 and 100. So it may be the 150 and 120, maybe. Okay. So if this is the 150 band, so and this is the number of genotype. So you can write in Excel this way. Here you type your SSR name, SSR01. Now the genotype name G1, G2, G3, like that. Now here put your band size 150 BP. Okay, you can count there is the 22 number of genotype. Okay, so this one is 22. Now 150 base pair. Where the 150 base pair number of one genotype? So you can put one here. Here 6 and 7. So 6, 1, 7, 1. Then in 16 number genotype. So 16 number genotype, put 1. And then 20 number genotype, 19 number. So 19 number, put 1. And then 21 number. So in 20 number, 1. Now the 120 base pair. So 120 base pair is 2, 3, 4, 5, 6. So 1, 1. Then here 1. One, and 1. The blank one is Otherwise, the blank. So blank one is you can replace with the 0. So this is the scoring of that marker. Okay. So this way you can score in binary score of the marker. Okay. So from the binary score, you can analyze the marker efficiency parameter. What are the marker efficiency parameter? You can see this one of the article efficiency parameter of SSR marker. So what are the parameter? Parameter means PIC value. We are know polymorphic information content, marker index, diversity index, resolving power. Sometimes it's also called the discrimination index. Okay, so we need this type of parameter for analysis or selection of the any molecular marker. So that's why we need to, and also we do some dendrogram like that from the or phylogenetic tree. Also, we can do the principal coordinate analysis PCOA from the marker. So that's why this is the marker image. So from the marker, how we can analyze such type of parameter. Okay. So for that, this is the marker. One of the SSR marker I have doing. I don't know whatever the marker name, but I just downloaded from the internet and then code it. You can also do your own gel image when you are generate the gel image in your lab. Then you can see your marker or gel image and then score like that. If you have any doubt regarding the scoring, please tell me. So I think no one have the any doubt regarding the scoring. Now we can calculate the marker efficiency parameter. So Chitrapal, okay. So this is the gel picture. This is one of the SSR marker, gel picture of SSR marker when we are doing gel electrophoresis with SSR marker. Okay, so we can found such type of gel picture. Now you can see the band size between the these are the band. That band size between the 100 base pair and 200 base pair. Okay, so maybe this is this band 150 or 180, whatever that. I am thought that this band, the upper band are 150 and lower band is 120 base pair. So there is the 1 to 22 genotype. So I have written here 1 to 22 genotype. Okay. Now the first number of genotype, which band is present? 150 base pair band is present. So that's why I am written here 1. And 120 base pair band is absent here. So that's why I am written 0. Next, in G2, there is no 150 base pair band, but there is 120 base pair band. So that's why I have written 120 base pair. Like that, I am scored all the 22 genotype. Okay. So this is the binary score. It's okay, Cipra? Got it? Okay. So now how we can score, calculate the marker efficiency parameter. Before this software, I don't know, very few person know about this software, very few person. Okay, most of the students are doing marker efficiency parameter. Mainly they are doing PIC value, calculating the PIC value with the help of the Excel. They are doing like p value or frequency. Then it's calculated with the formula in Excel, and they put their value in the paper. But there is a one online tool. That tool name is iMake. iMake. It is online tool, not required to install. It is online based tool. So you can found the link in article. This is the actual link. Okay, you can found. I will share this link in the group. Okay. So this is the interference of the iMake marker efficiency calculator. Okay, and you can very easily do your marker with this software. Okay, so first, all you are open this iMake tool through, like, type iMake online marker efficiency calculator. Then you can found different type of search result, but there is the one of the result in iMake online marker efficiency calculator PMC. Okay, you click on that, you can found one paper, and before the within the abstract you can found this link. When you click on that link, that interference will be open. Okay, now how can I calculate? For calculation individual marker, you need to separate the individual marker scoring. Here the marker scoring is like that. So we need to transpose that marker scoring this way. You don't need the genotype name. Here you can type 150 base pair of SSR1. So you can write like SSR, sorry, allele 1 and allele 2. Now save it in separate Excel. SSR 01. And ensure that there is no other sheet in that Excel. So there is the only sheet 2. There is no other sheet, and save it. Now go to your iMake tool. Our sheet is Excel sheet. Browse your data. Upload it. Now click on iMake. You can find all the result. This is the heterozygosity, PIC value, EMR ratio, heterozygosity average, marker index, discrimination index, and resolving power. Whatever I show you in your paper, in that paper, like that. Here, PIC, MI, DI, RP. Here you can find PIC, MI, RP, DI, and others also parameters. You can, that is the heterozygosity average. Okay. Now you can download the table. So in Excel file, you can find all the calculation. So one thing remember, you cannot analyze all the marker togetherly. Okay, you need to, if you have 20 marker, so you need to create 20 Excel, separate Excel, and individual Excel will give the individual marker result. So if you have the another SSR marker, so we can create another Excel SSR2. If I have the 20, then I have just created SSR1, SSR2, SSR2, 3, SSR4, like SSR20 Excel separately. And we can get result 20 Excel. Then we arrange this result in one table and you can put in your paper. So this is very easy, nothing to do. You just score the marker and analyze with this iMake tool. Any doubt? I can share the link into Excel, into your group. You can download it, or you can analyze through this. I have already shared in the group. So you can check it and do yourself. If anyone have any doubt, Abhishek Roth has raised a hand. You have any doubt? Mr. Abhishek Roth, have you any doubt? So I think no one have the doubt. Okay, let's we do some dendrogram analysis through the marker scoring. Okay, so already similar way I have scored another score with some population details. Okay. So we can just copy this, and so I don't need the populations. So this is this such similar way I have scored from 2, 3 different marker, and now the final score of the 11 genotype. Here the 11 number of genotype. Okay, now I can reduce 11 to 10. So this is 10 number of genotypes with 11 number of total allele of different 2, 3 marker. We have the 11 number of allele, and now I can analyze a phylogenetic tree with this molecular marker. Okay, so how I can analyze this phylogenetic tree with one of the very common software. Maximum people are known that software, that is called the Darwin. Okay, for Darwin, it is a free software, so you can go in Google, write Darwin software. So from CDAT, you can download it. So this is a download form. You need to your email ID and some basic information. Submit the form, and you can get one link in your email ID, and you can download and install in your computer. After the installing, you can found this type of logo. This type of logo. Okay. So this is a Darwin software. Okay. Now I am preparing the input file for Darwin software of this scoring. So I have the scoring, such type of scoring. For the Darwin analysis, we need to write like here, var, var, and row wise, you need to type your genotype name, whatever your genotype name. If here, this is the Ganga, Jamuna, Saraswati, whatever you can write. I am just putting G1, G2, G3. And there you need to write like this way, A1, A2, A3, or X1, X2, X3, number of allele, total allele. Okay. So now you need to save into another folder. The folder name is Darwin. Okay. And save it in text delimited. Text delimited. This so that scoring is saved in as a text delimited. I rename that scoring, that is named I just marker SSR. Okay, so this is the input file. Now you need to open the Darwin, and you need to import data matrix. We have the binary data matrix, so we need to import that data matrix. Desktop. training, Darwin, SSR, open, and save as. Then save and click on the OK. Okay, this step is over. Data is already imported. Now the dissimilarity analysis. For dissimilarity analysis, you need to choose calculated from single data. Okay, then click on the SSR. But now here you need to check this coding section. Coding is 0 equal to absence and 1 equal to present. When I am scoring, I thought 1 is the present and 0 is the absence. So here it is also click on that, click on there. So save that dissimilarity and press OK. It will take 30 seconds and it's over. Now you can go factorial analysis. Analysis, choose folder, open that, and save the coordinate, save as, click on OK. And you can get principal coordinate analysis. Okay, where now you need to select this one. Here you can open identifier file. You choose one DON file. It automatically create. Don't worry. Then you choose on the var. Click on var. You can found there the genotyping name are showing. Now you can click here and increase the size of your font size 12. Okay, so this is your grouping, and we can found there is the 4 group of genotype 1, 2, 3, 4. Okay, it's over. This is the factorial analysis. Now if you want to color the 4 group into different way, so click on that 25, write 25 here and choose the genotype name like G1 and G3, one group. So we click on G1, then click on control and click the G3, then choose your color, then press OK. It will be green. Now this one G7, G8, G6. So G6, G7, G8, G6, G7, G8. Choose another color. It is red. Then G2. Maybe blue. Click on. It will be blue. And the others. So other one is G10, G4, G5, G9. Choose purple color. Okay. So this one, the purple color. So this way you can changes your principal coordinate analysis. Now we need to analyze tree. So we are going to hierarchical clustering. Okay. Then choose your dissimilarity matrix. Change here UPGMA method and save the tree. Save. Okay. Now tree construction, you can click on that and you can found the tree, but we know that hierarchical horizontal tree. So this is the hierarchical horizontal tree, your phylogenetic tree. Now click here and you can found identifier. So select your var and you can found the genotype name. Now increase the genotype name. Okay, So you can see this is the two cluster. This genotype have the two cluster, and G9 and G10 is most of the similar genotype. Okay, you can see here. Now you can change this dendrogram into different. Before that changing, I am coloring that two group. One group is G1, G2, and G3. Okay, so I just give the green color and okay, and others G10, G3, G5, G6, G7, G8, G9, I am giving a red color. Okay, so I found two different cluster into different color. Now I am changing this dendrogram into growing root format. Okay, you can see this looking very nice. Okay, so this way you can save your dendrogram and put in your paper. Okay, for save, you can just click on that, save as tree, save as EMF graphic. So if you are record or save as a EMF graphic, you can found the transparent background, or if you save as tree, then you can just found as a image. So this way you can save this picture, okay, and create your dendrogram with this Darwin software. So this software is actually free, and you can also modify your dendrogram or PCA analysis. Okay, this is your PCA analysis from the molecular data. One another important things, sometimes we found some frequency distribution diagram for the molecular marker. So this is the bar diagram from here, and you can found this is the frequency distribution. That's why this frequency distribution indicated this is the genetic dissimilarity. Okay, this is the frequency of genetic dissimilarity, and this is how many number are belong or on that dissimilarity. Okay, so this way you can also represent the frequency, but most of the paper they skip it, but some good journal also showing this frequency distribution. If you know the interpretation, then it's okay. If you don't know the interpretation of frequency distribution, then skip it. Okay. So from the Darwin, we can found one phylogenetic tree, one PCOA analysis, and another one is the frequency distribution, the bar graph. Okay. So participants, if you have any doubt to operate this software, please tell me. Actually, this is a hands-on training, so you can also do when I am doing. Okay. Sir, can we do this with protein data? Yes, if you have the protein data, and that protein data you can convert it into the binary matrix, so you can create your own phylogenetic tree on the basis of protein data. Okay. Yes, thank you. Any other participants? Some participants have the unstable connection. Twinkle, any doubt?
Yes, sir. Actually, that time internet was unstable. I could not see how you have done. I have seen till where you have started downloading, importing the data. Till there only I have seen.
Pardon, please.
Means sir, I have seen my internet connection was unstable, so I have seen only till that point where you have went file and then import data. Till there only I have seen. If you can repeat once.
Import the data after. Yeah, so you need to repeat this process. Okay. Any other participants you need the repeat this process or any doubt? So I am repeat the same process again. Okay. So I close the software, everything. So first I have the score, and that score I have converted like this way, and I can already say that in the first row, first column, you need to write the VAR. You can write anything here, but var is the one of the code that Darwin received. Okay, and this way you need to write column wise. You need to write your allele. Okay, and row wise you need to write genotype name. Now save it into the text delimited file like this. Text delimited. You can see this text delimited. Okay. After save, you can found this type of text file, and other file whichever already generated. I am just deleting. Okay. Now you can you open the Darwin software. This is the Darwin software. Go to File, Import Data Matrix. Select your data, open, and save as data. Okay. After that, click OK. Then you need to dissimilarity file calculated from single data. Okay. Choose your data, SSR var. Then check here whether it is click on there 0 equal to absence and 1 is equal to present. Okay. And here the method is present and absent. Then save dissimilarity as. Here save option, you can click on save option. Then you can found here the OK is visible. Then click on OK. Wait for 30 seconds. Okay, click on OK. Then go to the factorial analysis. Analysis. Choose your disk file, open, and save coordinates. Save and click on OK. You can found your principal coordinate. Okay. Now we need to analyze tree. For tree analysis, hierarchical clustering. Choose your dissimilarity file. Click UBKV method, save tree, save, and click on OK. Then we need to construct the tree. Draw option, select draw option. You can found the phylogenetic tree. Now we change it phylogenetic tree, hierarchical. Now you can see here the no genotype name. For genotype name, you click on this identification and illustration. Click on here, you can found one question mark sign. Click on select identifier, select the DON file, and then again click here and you can choose VAR. So you can found genotype name is coming. Now you need to increase the genotype name size. Now you want to color, then you choose the different group genotype with the pressing control. This is red and another so you can see two cluster are here. Now you can change if you want growing root method, if you want hierarchical vertical method, if you want radial method, you can also axial method. So different type of view are here. So whatever you need, you can choose. Okay, and save it as a EMF file. Okay, save tree as and record EMF. Whatever you need, you can save this one. So this is the phylogenetic tree analysis, and this tree analysis can be done with the SDS page scoring for protein, or if you are analyze different biochemical or isozyme marker through SDS page, you also or page polyacrylamide gel electrophoresis you can do, or if you have any binary matrix, you can analyze such type of dendrogram through the Darwin software. Okay,
Okay, sir. Thank you.
any doubt? So next we go for the population study analysis. Okay, so for population study analysis, first you understand what is the population. Population means in certain area and certain time, if you are founding some specific organism, so that specific organism in a certain area for certain time, specific organism whatever present, they are called the population. So for example, West Bengal have some population, Assam have some population, Tripura also some population. But if you are count in a specific time, so whatever some population are all time moving from Assam to Tripura to West Bengal. So that moving population you cannot count. But if in a certain time you fix 3 states, no one can moving, but those are moving into West Bengal, they are count as a West Bengal population. Those are moving West Bengal to Assam, that are count as a Assam population, but cannot move after that. You cannot, we cannot move from one to another. So we choose some number of population from the Tripura, we choose some population from Assam, and we choose from some population from the Tripura. Now we get 3 number of population. Now we can test whether this population actually vary to each other or they are admixture. Means most of the population are 50% population Tripura are going to West Bengal and 50% population go to Tripura. So there is admixture. So you can differentiate between the or among the population through the molecular marker. Okay, so that's why I am just showing you an example of the scoring with same whatever I have created some phylogenetic tree. Okay. So same population here I am showing, but I have put one column. That column name is the so here I am giving one of the line, that is population information. Okay, so first one is the genotype information, second one is the population information. So population information is West Bengal, WB is West Bengal, and MH is Maharashtra. So we can see here the G1, G2, G3 and G4 coming from the West Bengal, and G5 to G11 is coming from the Maharashtra. Whatever, I am just delete that last one for because the allele number and genotype number are same. So that's why I'm deleting this for your better understanding. So now this is the our information. So here the molecular marker information. Here I am giving the population information. So when you collecting any genotype, your always you put your information, keep your information whether that genotype where it is originated. If you collected from West Bengal, but what is the actual origin of that genotype? Maybe it is originated IRI, maybe originated in Punjab. So that's why all time you need to keep your record of every genotype or every isolate in case of pathology or microbiology, isolate where you are collected that genotype or isolate. And your population, you have the 30 genotype. So among the 30 genotype, you can found there are 2-3 population. That means they are coming from the 2-3 different places. Okay. So that's why you divided your population, your isolation or your collection into different population. So I have collected and I have recorded this population and arranged this way. So for the population study, you need not extra software. One of the software you need, but that software will be integrated with the Excel. So that's why you don't worry, and that is also the free software. Okay, I am showing all time free software. Okay, so now the arrangement of the data, how you can arrange. Clearly, you can see there the first column. There is the genotype name, then second column is population, then number of allele and their score, individual genotype. Okay, and first one is the number of allele. Remember, this column is number of allele, that is 11. Second column is number of genotype, that is 10. Third column is the number of population. How many population? West Bengal and Maharashtra. So population is 2. Now in West Bengal, how many genotype? 4. In Maharashtra, how many genotype? 7. And forgetting. Okay. So this is your data for population starting. Now you need another macros. So for that macros name is GenLX. GenLX software. Okay. GenLX. You can download this GenLX software. So download is over. Now extract that zip file. After extract, you can see GenLX 6.5. Go to that folder and you can found one Excel file. Just click on that, double click on that Excel file. Okay, so at this moment, this file not support my computer, but I have already another GenLX. Yes, so we can, yes. After clicking on that, you can found one option, enable the macros. You can just click on that, enable the macros, and you can see one of the option here, like GenLX with in your Excel. So this is my data here. I'm just click on GenLX, and now I'm going to frequency analysis. For frequency analysis, you need to click here. After you can see the number of loci is 11, sample number 10, and population is 2. Everything is okay. Now the matrix all time binary haploid. When you are scoring in 0, 1, it will be the haploid format and binary. So binary, you can choose binary haploid and click OK. The sample does not equal sum of population size. Okay, so I am one mistake, that is 4 and 7 is the 11, but here the 4 and Maharashtra have the 6 population. Okay, now you can click on the frequency. Okay, yeah, you can get this option. Okay, now you check all and click on OK. And automatically it will be analyzed. So two important file, there is lots of file generated. So one is the important file is the frequency distribution, that is the different type of number of different alleles, number of affective alleles, Shannon diversity, diversity index, unbiased gene diversity index of West Bengal population. So population wise, you can found the different diversity index, and also, and this is the total. So for West Bengal it is, for Maharashtra, for total you can see. So you need to the BAFP file. Now another file is that is BAFL. Here you can see any one of the population, there is the any unique band or private band, which band are common, which are the private bands, means unique bands, you can see here. And this is the their bar diagram. So for this analysis, you can found the frequency distribution and different type of the population. For population study, it will be required. Then we need the genetic distance. So for distance, we need the genetic distance, and we click on the binary haploid and output total distance. Click on OK. So you can found the GD, genetic distance. So this is the genetic distance, but we don't need this genetic distance at this moment. But for the genetic distance, we can analyze PCOA. So analyze the PCOA, color the code of the population, individual population will be colored, and you can see this is the PCOA analysis. So where your genotype, you can see West Bengal genotype are red color here and Maharashtra genotype here. But you can see one genotype red color is in the Maharashtra population. That's mean that G4 actually the West Bengal population, and they migrated into the G4. Okay. So this way you can identify which genotype actually were situated. Okay. So this is the principal coordinate analysis, and you can understand the structure or population about the different genotype. And now we heard another analysis of molecular variance. So that's why you go to that distance option and click on AMOVA, and you can found AMOVA analysis. Okay. So this is the analysis percentage of molecular variance or AMOVA. And you can see within the population, 61% variation, among the population, 39% variation, and this is the AMOVA table, and you can found the p-rand value is 0.005. That means it is the smaller than 0.001. That means the population will be significantly varied. Okay. So this is overall the population study. Okay. Using the molecular marker and using this free software GenLX. Okay. So I think you got my point. If you have any doubt, please tell me. So participants have no any doubts. I think they are thinking this is a very tough thing, but it's very easy. For that, you can refer this paper, a comparison of efficiency parameters of SSR markers and genetic diversity in amorphous, Pionephilus. You can found different types. It's also in Moonbeam, you can found similar type of paper, but this is my paper. You can follow this paper for interpretation. Okay. So next we go for the next software. Okay. And son of Nag has raised. Do you have any question? Chanok?
No, sir, no, sir.
So why you are raising your hand? So then we go for the next software. I don't know this software, whatever previously discussed or not in this training. Mega software, MEGH, Mega software, this already done or not? For phylogenetic tree analysis. Okay. So now we can do some analysis with mega software. Analysis means phylogenetic tree. So for that we need some sequences. Okay. So all we know that we are doing most of the cases, those are from microbiology. They are doing their sequencing, Sanger sequencing through different companies and get that sequences. But after that sequence, they don't know whether these are varied or not. These sequences are within. If you have the same type of isolate and you get the sequence of that 10 isolate or 5 isolate, okay, after that you want to analyze or create a phylogenetic tree with your own sequences. One of the phylogenetic tree you can generate it through the NCBI, but it's have your only one sequence and other sequence of the different population. But if you want to know whether your sequence, within your sequence, what are the genetic diversity, if you downloaded the different type of protein sequence and you also want to what are the proteins, protein sequence, what are the diversity within the protein sequence, you can need to mega software for analysis. Okay, so let's start that mega software. For that mega software, you need to go to the Google and search mega software. And this is the mega software. You can go to the download section and download and install it. Okay, you can download it and install it. Okay, some question are there. You just fill up this question and freely it will be downloaded. After the install, you can get one shortcut, and that software like this way, this mega. Okay, now if I just minimize this software, and for the analysis we need some sequences. So I have collected some sequences. I have already some sequences. So there are ITS based some sequences of bipolaris sorokina. Okay, so this bipolaris sorokina, there are 4 sample, 1, 2, 3, 4. Okay, so this is the isolate name, and this 4 sample we want to create a phylogenetic tree and distance whether this genotype are same or not, or this isolate are same or not. If not, whatever the distance between among the sequences or isolate. So close this file and you need to open. First, you need to import your data. So open a new session, file session. Sorry. Edit or build alignment. Create a new alignment. Click on OK. From DNA sequence, go to edit and insert sequences from file, and that file into the desktop training mega and SB text. So click that and right click and delete the last phone number. So there is the phone number of sequences. Now you need to save these sequences. Okay, so export the alignment as a mega format. Okay, and save that alignment in the same folder where the mega folder is. Okay, you need give the name new SB like that. SB, put the title. Okay, protein coding nucleotide sequence of data. If you know that this sequence are protein coding, then you click on yes. If you don't know whether they are protein coding or not, then click on the no. So I have no information whatever they are protein coding or not. So that's why I'm click on the no. So close this alignment. Okay. Now you want to analysis diversity or phylogenetic tree. So go to the construct maximum likelihood tree. Click on that and select your folder. That folder is the training. Choose the folder and click on open. So here you can change your method. That is the we are always using the default method, that is maximum likelihood. Okay, and complete deletion. So there is nothing to change. But if you want to bootstrapping method, so that time you can click here. It by default it will be remain 1. If you want, it will be the 10, or it will you can increase up to 100,000, 10,000, whatever you require. I have just putting 10 and click on OK. From the file, and that file, sorry, just I am press 1. Okay. So from the file node, random tree active data, and you can get your phylogenetic tree. Okay. For phylogenetic, after creating this phylogenetic tree, you need to click on OK. Okay. So you can final your phylogenetic tree this. Now you need to save your phylogenetic tree, export current tree, new quick. Okay. This folder including branch length and bootstrap value. Okay. And save this folder is B tree in the same folder. Okay. So from Mega, you can found this type of phylogenetic tree. Okay. And you can change this phylogenetic tree into different way. Okay. There is lots of option. Okay. But I cannot prefer this option like circular. Okay. But I cannot prefer this option from the Mega software. After getting the NIC file from the Mega software, now you can get that NIC file. This is the SB tree is a NIC file. Okay. From that, we are using another different software. That software called is iTOL. And from iTOL, we upload. This is also free online tool. And we just choose that SB tree and upload here. rectangular. Okay. Now you can see this is the your dendrogram. Okay, and if you want to rectangular way, so this way is the rectangular. If you want to circular way, so you can see this is your dendrogram. Now, what is the importance of this tool? You can select the branch and click on that and label as you want this type of color. Okay, you can choose label. You can choose here label. Another one is choose label. So you can choose different color, and you can also change that length also. Okay, this also different way using this software. After that, you can go export, and you have the different type of option for select your file like PDF, EPS, or PNG. If you want to PNG, then you can choose your DPI size. If you want to 600 DPI, whatever which is mostly required, so then you can export this 600 DPI. So you can get high resolution dendrogram from using the Mega followed by iToll software. Okay. So this download is completed. So you can see this is the, now you can see that this is black because the figure actually in transparent background. So in PPT, any one of the PPT, you can see this is your figure, and this is your figure. Okay. So this is all about the Mega software followed by iToll. Now, if you want to make the distance, genetic distance within the isolate, you can do with the Mega software. So let's tell me if you have the doubt regarding the Mega software. Those are working on protein sequence, similar way, you can save your protein sequences and then you can import in Mega software and you can get the results. Okay. For distance analysis, go to distance, compute pairwise distance. Click on OK and you can get this distance. So SB101, SB06, SB07, SB08. And this is same SB01, SB06, SB07, and SB08. And this is the genetic distance between the isolates. Okay. So this way you can also analyze the genetic distance also. Okay. So whatever you need, you can analyze through the mega software. Basically, it will be the genetic distance and or diversity analysis within the sequence. Okay. That's why software name is molecular evolutionary genetic analysis. Okay. From that, you can analyze everything regarding the molecular. So if you have any doubt, participants, have you any doubts? So I think no doubts. So I'm closing that software. So Sanab, Shanab.
Yes, sir.
So tomorrow I'll take class, na?
Yes, sir. Yes, sir.
Okay. So today I'm closing here. Okay. If participants have any doubt, can ask in group. Okay. So tomorrow we'll discuss about the different bioinformatics software regarding the genome sequencing. Okay. And we can see how we can create the chromosome map, or how we can analyze the different gene structure using the bioinformatics software, and also we can analyze the genome-wide analysis through the bioinformatics software. Okay. So thank you, all of you.
Okay, thank you, sir. Thank you for taking this wonderful session. And now also we are closing the session. Thank you to the participants. If there is any query, you just post to the WhatsApp group. So in next sessions, I will definitely answer on it. Thank you.